here, and this is still on topic 1. We're talking about cell surface area to volume. This is one of the topics on topic 1 that I think that a lot of students have a hard time grasping the concept of it, and I'm going to try to make it as simple as possible for you. So we have one understanding for this topic of surface area to volume, which is cell surface to volume is an important limitation to cell size. We're going to understand what it means that it's an important limitation. What does that mean? Now, through that, we have these concepts that we're going to try to explain the relationship between the cell size with the surface area and volume ratio. We're going to say why the cell has a limited size. What are the adaptations we can to maximize the surface area to volume ratio? And what exactly is happening in the surface area as well as the volume of the cell. So all of these stuff is in the next slides. So the biggest question you have to ask, why cells don't grow to large size? Of course there is exceptions, but why is it that cells, when they reach to a point, they have to divide? That's what this whole next slide is going to be about. We're going to understand why the cell must divide and not continue to grow. So it's all about surface area to volume ratio. Now, students will see the surface area to volume ratio and start thinking, holy shoot, we are talking about math and I suck at math. Well, let's make it into biology words. We have the word surface area. When we say surface area in bio, we're basically saying the cell contact with the outer world. Or should I say with the outside? That means how much can the cell be inside of the cell be in contact with the outside, okay? And volume, we're going to talk about the space inside the cell. The space inside the cell. In this case, we can say the cytoplasm. So we're talking about the contact of the cell with the outside in relationship to how much space there is inside of the cell ratio. So this is what it is. So moving on, let's focus first on just the volume. When we're talking about the volume here, we're talking about the cytoplasm. The cytoplasm, it's all this inside of the cell. Now you see that this picture right here, it's in 2D. So to make it more understandable, let's look at the 3D view, like these two boxes right here, box A and B. Now, you see the box A, you can fill up more boxes inside the box A than you can do it on box B, which that means box A has a bigger space, or should I say a bigger volume compared to box B. That means you can fill up inside of the box A a lot more. In box A, there's more cytoplasm. In box A, there is more space inside. That means box A has a bigger volume. So we can say volume here, it is big compared to this volume here, which is small. Now, is this a good thing that box A has a bigger volume compared to the box B? Well, all of this depends on the contact with the outside. What does that mean? Well, first let's understand one thing. It's great if you have so much on the inside, but if the inside doesn't have enough contact on the outside, that could be a huge problem. And why is that? Because here's what happens. You always want to get rid of toxics, right? And if you don't get rid of toxic on time, you can have metabolic disruptions. That means something can go wrong inside of the cell. What does that mean? Let me give you an example. So here we have 3 cells. And this little dot right here that you're going to see, this dot, it represents a toxic. It's a toxic material, okay? So to go from the middle of the cell, right, they have to swim through the cytoplasm to get out, right? Let's say this takes 1 second. Awesome. The toxic left the cell in 1 second. In this cell, the toxic left in 5 seconds. And then this toxic in this cell left in 1 hour. Let's just say that the toxic can stay inside your cell for max of 10 seconds. After 10 seconds, if this toxic, okay, does not get out of the cell does what? It dies. It automatically shuts down. Believe it or not, even cells can suicide. If this cell feels like, oh my God, I have too much toxic stuff inside of me and I can't get rid of me, what I do, I just kill myself. Cells do that too. There's even a scientific name for it, which is called apoptosis, but you don't need to know this. I'm just giving you a little insight here. So cells, they need to feel that they're always in balance. But if they have too much toxics inside of it and they can't get rid of it, they actually kill himself. So if we see that the maximum amount of time you can hold a toxic is 10 seconds, then after 10 seconds, what does this say about this little guy here? If it took 1 hour to get rid of this guy, what would the cell do? Kaboom, dead. It will kill itself. Because 1 hour is too long for this guy to make it strip through the entire volume, the entire cytoplasm, to eventually get outside, be in contact with the outside. 1 hour to get in contact with the outside. The cell will not function that way. So, nope, you do not want to grow this big. So what do? You stop right here. See, up to here, the 5 seconds was okay with them. But once you get to the cell becomes bigger, that means the volume inside is getting bigger. What's happening? The cell was not going to be capable of getting that toxic on time. So instead of getting bigger into this huge guy, which that's not the way to go, oh, let's divide again into 2 small cells. Oh, and these 2 small cells will go back to me that 1 second to get out. That's why the cells do not get so big. Because if they get too big, then they can't handle the toxics inside. They will die. So every time they reach a certain size, it's time to divide. Because you want to keep the cell volume always small. Let me explain this and how is the volume actually related to the surface area. Let's look at the surface area. This means contact with the outside. That means how much contact? Look at these 2 cells. You have cell number 1 and cell number 2. As you can see, number 2 has more contact. Number 1 has 1, 2, 3, 4, 5, 6. You can even put a 7 here and an 8 here, but that's pretty much it. But the folding allows you to have more contact on the inside. Look, it's inside from here, inside from there, inside from here. So number 2, this guy has a lot more surface area. He has more contact with the outside. What does that mean? That means this guy, all these little things that are inside the cell has more contact. So imagine this cell here, right? Let's say this is a toxic right here. For him to go contact with the outside, he needs to do all this work to get out. Agree? Well, if I did this, if I had this little toxic here, but instead I folded this like that. Imagine I folded. Look what happens now. The beauty of it, now that you fold it, you just need this tiny little motion to get out of the cell. So the more folding there is, the more surface area, the more contact you have with the inside. So it's all about how much contact the cell has with the outside. That means every single part within the cell should be quickly to have a contact with the outside. So what's really happening is that the cell has a certain communication going from in and out, right? So this particle here is closer to the surface, so that means it's closer in contact with the outside. But then the particles that are right here in the middle, they are not quickly in contact with the outside, so they need a little bit extra work to get there. So this is the surface area. Right here, this is the membrane, this is the surface area. This is what is in contact with the outside. So as long as you're small, that's fine, right? Because you're going to be quickly moving out. But if you get really big, these particles here will be quickly going out. But the particles right here in the middle, they're going to need a lot more time to get out, so they have less contact with the outside. This is the reason why I want to make it smaller. So here's an example. You have here, which we call this a nerve cell that you have in your body. And you have a normal cell here. It can be a liver cell. So look at this two. Which one has more contact? If you actually do the line up here, this is the contact with the outside. right? The blue line, which is the plasma membrane, that's the contact. So who's more closely to contact? These guys, they are more close to the contact, the red dots. But if, let's say, let's use another color here, let's use green. But the dots that are here, these toxics that are here, they are not in contact with the outside. They have to walk through to get to the corner. But imagine here, on this one, you have all the contact here. Look, all of them are very close in contact with the outside. So that means which one of these two has more surface area? Obviously the nerve cell. It has contact from every single area of the cell. This is the entire cell, by the way. All of this, you can see this is the nucleus right here. All this is contact, contact, contact. So the more folding you have, the more contact you have. This, only the edge has more contact with the outside, but inside the volume, right in the inside, barely has contact. So this is another example of a disease called the celiac disease, which here, for example, is the wall of your intestine. Here, the wall has these foldings, which allow you to have more contact, while here you have tiny little foldings. So imagine, here you can have a lot more contact with the outside, while here you barely have it. So the more folding, the better it is. And when I say more folding, we're talking about surface area. So let me give you an example here. Okay, this is just to like solidify what I'm talking about. So imagine you have this circle right here. Okay, and we're gonna have all the toxics here, and we're gonna have another color toxic too. We have toxic here, and we have another color of toxic here. Right? Now, who is more in contact with the surface area? Of course, we're using black here. You see the black, this is the surface area. This is the plasma membrane, right? This is actually in contact with the outside. Contact with the outside. And then all the space inside, as you can see, all the space inside here, this is the volume, or this is the cytoplasm. Now, out of the cytoplasm, what is more in contact with the environment? Obviously, if I highlight it here, this area right here is more in contact because they're closer to the surface area, or should I say the plasma membrane, so they're more likely to go. Who's not in contact? The inside. That means this area here barely has any contact with the outside. They have to do a little bit of movement towards the edge in order to get out and be in contact with the outside. But if I decide to draw another cell, and look at this cell now. If I do this cell, it's the same size as the other one, but this time I'm folding it. Okay, it's the same cell, okay, but I just folded that cell. And you're gonna have the same toxins going on, right? And you're gonna have the other one, which basically was the yellow one. Let's put the yellow here, and we had here. Oh wow, look at them. They're also close to the edges. And then you have the blue ones. Look, they are also very close to the edge. That means they're close to the plasma membrane. Now, which one of them are closest? All of them. In this case scenario, if I highlight this whole thing, look, you even touch the yellows and some of the blues because all of them are very close to the plasma membrane. So they are very close in contact with the outside. So which one has more surface area? In other words, which one has more contact with the outside? This one, of course, this guy right here, oops, he has a bigger surface area than this guy. Why? More folding. It's not because he's bigger. It's just because there is more contact with the outside. They both have the same volume, right? Here, the volume is the same, and the volume here is also the same. The only difference is the surface area here, it is small, and the surface area of this guy is big. So which of these two are the best one to work? Cell number 1 or cell number 2? Obviously, cell number 2 would be the best one. Why? Because the surface area, it is big to a volume that is small or the same. This one, you can see that the surface area is too small for the volume. So it's always about making sure that the surface area, it is, so I'm going to put here, surface area always have to be bigger than the volume. It is extremely important that you know this. The surface area always has to be bigger than volume. In other words, the contact with the outside should always be more than the volume that is inside, or the space that is inside. It means everything inside should be in very close contact with the outside. So the more there is contact with the outside, the better it is. But if you have too much things inside that are not in contact with the outside, that's bad news. So it's always about more contact compared to volume. If you have too much things inside, in other words, if you have too much volume, but the surface area is too small or the contact with the outside is low, the cell will burst, the cell will be destroyed, and will not function. So there's an experiment that you do in class. Some teachers do it, some teachers don't. But this experiment is a perfect way to illustrate what I've been telling you this whole time. So what they do here is they get agar and they create cubicles, like these little squares right here, right, of different size. So there's 4 different squares or cubicles. And what they do is they throw it inside a tube that is made of HCl, hydrochloric acid. So this is all HCl. And what's happening is they boil this tube. Now, the HCl is going to start coming into this cube. So you can see here in the cup, right? You throw the cube into the HCl and then you take it out. When you take it out, these 3 cubes, look at the small one. You see, the small one had more contact with the outside, so that means the outside has touched all of it inside because there was more contact. The second one, you can see it. The only much contact was the edges. What happened to the middle part? Nothing. The middle part did not have any contact with the outside. And you made an even bigger cube. Look at the bigger cube. Even more. You can see there's even, even less on the inside. Look how white it stayed. There was not much contact with the water. So the bigger it is, the less contact there is with the inside. So between these three, which one was the best? Always, of course, the little one. The smaller the cell, the more your inside of the cell will be in contact with the outside. But if the cell gets too big, only the edges of the cell will be in contact with the outside, while deep inside of the cell will not be in contact with the outside. So out of these 3, which one has a bigger surface area? Obviously the small one. This one has the biggest surface area. And this one, surface area, is the smallest. And again, when I mean it's not because of the area, it's the contact with the outside. The cell, the smallest one had the contact, all the inside of the cell had contact with all of the outside. But the bigger cell, only some parts were in contact with the outside, the rest wasn't. So if we take this into a math consideration here, look, we start with the tiny little cell. Let's say this is a tiny little cube, okay? And putting here, let's look at the surface area. The surface area here is the contact with the outside is 24. And the surface area here is 96. And the surface area of this one, it gets 216 and then 384. Okay, the cell is getting bigger. There is somehow contact with the outside. But that's the edges of the cell. Now look at the volume. The volume here is 8. The volume is getting bigger. Volume means the stuff inside, how much stuff is getting inside. It's getting bigger and bigger. Now, when we look at the ratio of the volume here, when we look at the ratio, volume area to volume is basically 24 divided by 8, 96 divided by 64, 216 divided by 216, 384 divided by 5, 112. That's where you get the ratio. So surface area volume here is 3, 1.6, 1, 0.5. So if the teacher asks you, do you want the ratio to be big or small? Like, which one is better for the cell? For the cell to have a surface to area ratio. But let's put surface area ratio to volume to be big or small. Your answer is you want it to be big. You want it to be the biggest. The bigger, the better. The ratio, again, the ratio means the relationship between the surface area to volume. You always want it big. The smaller the ratio become, the worse it is for the cell. So at the end, you always want the surface area volume to be bigger, right? So the surface area always has to be bigger than the volume and not the opposite. As you can see here on the drawing, just to show you here a little bit on the drawing, look at these two drawings right here. This is one big cell, and you can see that it has a low surface area to volume ratio. These are 4 cells who has kind of the same size as this guy, but they're 4 different cells. That means there is more contact on the inside. See, you can see from here, from here, from here, from here, from here, from here. There's more contact. So that means it has a higher surface area to volume. So what does that mean? More particles are exposed to the outside. There's more contact with the inside. So this is what you want. So for example, you have this one big cell, or you have this group of cells that has the same size as this one. Which one is better? Obviously this one. Why? Because each cell has more contact. It's like the different cubes, they have contacts from different area. So when we wrap this up, we gotta make sure that the surface area to volume ratio, this guy influences the size of the cell. It is very important. The most important thing is that the surface area here always has to be bigger, way bigger than the volume. Not the other way around. As long as the surface area is bigger than this guy, you're going to have nutrients come in and out the waste with no problem. Okay? So the more surface area per unit of volume, more materials go out of the cell. It's all about having bigger surface area to volume. Okay? Never the opposite. So problem with why is it that we don't go to grow such large cells? Because the surface area in real life is always growing slower than the volume. The volume, that means the inside of the cell is always getting bigger and faster than the surface area. We don't have, in the normal life, we don't see the surface area growing at the same pace as the volume or even faster than the volume. It's very rare. It's always the opposite. It's always the volume. It's always the inside that is building up so fast that the surface area cannot keep up with it. And this is why the ratio becomes so small that that's a sign, oops, okay, the surface area volume is getting small. That's not good. What do? Divide. Let's divide the cell into 2. Because when we do this, we're going back to a ratio where the surface area is big again. So it's extremely important that you have a surface area to volume ratio that is big. The ratio should be bigger. The bigger the ratio, the better it is. The smaller the ratio, that's a bad sign. Division, division, division. Go back. Because also, you want the cells to be able to release that heat. Imagine if there's heat inside the body and you don't release. What's going to happen? The temperature is going to rise and you're going to burst. You're going to be melted. So you want that heat to leave the body as fast as possible. The best way to leave is if you have a lot of contact with the outside. That means you have a bigger surface area. So that's why the cells don't grow so big. Because again, you gotta make sure that the ratio is balanced, where the surface area is always bigger than the volume. So that's why you have all these large animals having many cells and not large cells, because you want these cells to function. You want them to be able to exchange nutrients and toxins in and out in a fast pace. Now, if you still want to have large cells, the only way you can do this is if you make them very long. So for example, you want the cell to become bigger and bigger and bigger. Jesus, that's not good. That's not going to happen. So what do? You have a better way. You still want to make it big. Okay, so if you want to make big, what do? This is a big one. Look, It's one cell only, but it's so big and thin, and voila, look at the best part. Contact. Everything is in contact with the outside so quickly. So yeah, you can have a very large cell, but only as long as it's very thin on the inside. That means the volume is low compared to the surface area. See, you have a large surface area in this picture and the volume is very small. Only this way. Or if you fold a cell like this, that means you have so many foldings, that means you have so much contact with the outside. But you cannot have a huge cell like this because, dude, what happens to these guys in the middle? They don't have contact with the outside, the cell will die. So, just to wrap it up, it's, again, the surface area needs to be big, while the volume should be small. And when this happens, the ratio will be big, and that's good. That's really good for the cells. However, if the surface area it is small and the volume is big, the ratio is small. And that means that the cell is dead, the cell will die. Not a good thing. So always go for a bigger surface area. Always go for more contact with the outside compared to the volume or the stuff that is inside. That's the most important thing. And that means it is the ratio between these two that controls the size of the cell. So, as we said, so here's a sentence. The smaller the surface area to volume ratio, more likely the cell will have to divide or it dies. And the bigger the surface area to volume ratio, the cells are small, and this means they are functioning normal. So the equation is always surface area over volume. Remember that. And this side always has to be big, and this side always has to be small in order to get a good result. And that's how you control the cell size. So here is a wrap up. You'll be able to talk about the activities of the volume and the surface area. Well, the activities is things coming in and out of the cell, keeping the control, getting rid of the waste, bring the nutrients. Also, the relationship of the cell size. It basically is the cell size all depends on the surface area volume ratio. It always has to be a big ratio in order for the cell size to be okay. If the ratio starts to get smaller, that means the cell size is getting bigger. And that means you need to divide. Why is that? Because otherwise you're not able to get things out of the cell quickly and they will die. Adaptations would definitely mean you having more foldings. You make more foldings, you get very thin. So this entire video has responded to these guys. If you need more questions, you can always ask me. But again, this volume to the surface area to volume ratio is well explained, and I hope you understood it. Thank you.